Let us look at the major types of methanogenesis reactions. The first one is acetoclastic methanogenesis. Here, the conversion of acetate into methane and carbon dioxide happens, and the predominant in many anaerobic environments. So this reaction is very important in the anaerobic environment. And the second one is hydrogenotropic methanogenesis. This is the Wolff cycle is a methanogenic pathway used by the archaea. The archaeon takes hydrogen and carbon dioxide and cycles them through various intermediates to create methane. This is common in various anaerobic settings. So this is the second type of reaction. And the third one is methyl-based methanogenesis. This includes methyl dismutation reaction, that is conversion of methyl compound into methane and other product. The second one is methyl reduction. So the reduction of methyl group to methane also happens. So the recent studies had highlighted the importance across different environments. So this is a very recently included mechanism. Let us look at the acetoclastic methanogenesis. The reaction is formic acetic acid is given here. This is converted to carbon dioxide and methane. So this is the reaction occurring in the acetoclastic methanogenesis. So which organisms do this is basically methanosaccharinales, that is methanosaccharina, methanosata. So these are all the two organisms which carry out this particular type of transformations. What is the characteristic of this particular process is a type of fermentation, but it differs from the typical fermentation pathways. So we had already seen what are the comparison between methanogenesis and fermentation. So this is similar to fermentation, but it is not actually fermentation. The ATP synthesis is based on the electron transport linked to phosphorylation, not substrate level phosphorylation. So that is the main characteristics of this acetoclastic methanogenesis mechanism. And this is most active in freshwater sediments, anaerobic digesters, and acetate contributes roughly 2/3 of the total methane formation. So we will be looking at these reactions. The second one is hydrogenotropic methanogenesis. The reaction is given here. A couple of reactions are given. The first one is hydrogen reacts with carbon dioxide to give methane and water. The second one is formic acid is getting converted to methane, carbon dioxide and water. The third one is methanol is getting converted to methane, carbon dioxide and water. So these are all the three different reactions occurring in the hydrogenotropic methanogenesis. That means hydrogen is basically involved in these reactions. And the characteristics is this occurs in all 5 orders of methanogens, formally a type of respiration type reaction. So that's the reason we did the comparison between methanogenesis, fermentation and respiration. This can grow autotrophically. These species can, these microorganisms can grow autotrophically. They lack an ordinary electron transport chain. So that is how this reaction actually proceeds. And it utilizes basically some special type of enzymes. One is F42O and another one is coenzyme M. And when we look at the energetics, theoretically this is favorable, but the practical growth rates and yields are lower than predicted. So this reaction may happen but may not give enough yield. And autotropic growth requires some substrate for the carbon assimilation consuming ATP. So these are all the energetics involved in this particular transformation. The next one is metallotropic methanogens. So the organisms involved include metallotropic methanogens, which are the subset of methanogens which are archaea. They belong to the classes as given here, methanomicrobia, within the order of methanomicrobiales. And what is the characteristics of this particular process? Basically, they metabolize substrates such as methanol, methylamine, DMS. And the morphology is these compounds can vary in shape from cocci to rods and may form aggregates or clusters. And what are the activities? Basically, the role in the biogeochemical cycle is they contribute to the carbon cycle by converting organic carbon into methane. This influences the global carbon cycle and contributes to the biogeochemical methane emission. So this is the major type of contribution from metallotropic methanogens. What is the energetics involved? This process generates energy which is a form of anaerobic respiration, and in the case of electron transport, the transfer of electron occurs through coenzyme M, coenzyme B and F42O cofactors. In the beginning we talked about this methanogenesis reaction happens when the sulfate is depleted. So let us compare this with the sulfate reducers. So the methanogens are inferior competitor for substrate, that is hydrogen and acetate, compared to sulfate reducer. So when a sulfate reducer is produced, they will first carry out the reaction and not the methanogens which can carry out the reaction. So that is the major difference. And when sulfate is present, methanogenesis is typically low, and in the sulfate depleted anaerobic habitat, so this is where actually the methanogenesis happens, especially freshwater sediments, sewage digesters, rumen, some marine sediments. The methanogenesis play a central role as hydrogen scavengers and terminal mineralization of acetate. So we will be looking at these reactions. And these compounds generally contribute significantly to the carbon cycle. And here is a network of decomposition of soil organic compound in the high Arctic peat soils. We have plant polymers which are getting degraded. We also have microbial biomass. So the microbial biomass can degrade proteins, peptidoglycans and compatible solutes. And the plant polymers, especially the polysaccharides, are being degraded. So these are all the various reactions by which, see, the end product is the methane. So we have polysaccharide, we have protein, we have peptidoglycan, we have compatible solutes, all these things are there. They undergo various types of reactions. Say for example, they may be A here given is a hydrolysis reaction. So the polysaccharide on hydrolysis gives sugars. Once the sugar is formed, they undergo the reaction B or D. B, C, D are primary fermentation reactions. So the sugar undergoes fermentation reaction to produce ethanol. And once the ethanol is produced, then it undergoes the next reaction, which is E, that is ethanol fermentation to propionate and acetate, or to acetate and hydrogen. So that is what happens. So ethanol is now getting converted to propionate, and this undergoes the reaction I. What is this reaction I? The propionate fermentation to acetate formate and hydrogen or acetate. So this leads to formate formation, and the formate now undergoes a reaction called M. M is nothing but methanogenesis from formate. So that leads to methane. So this is how polysaccharide can be degraded to methane. Similarly, we can look at the proteins and the peptidoglycan. They undergo reaction N and O. So what are N and O? Proteolysis and pentaglycine proteolysis are the two reactions that leads to formation of glycine, which is the simplest amino acid. And this undergoes various reactions like P, Q and R. The PQ is glycine cleavage and degradation, that is the first stage. Then sarcosine reduction and betaine reduction. So all these three reactions occur that leads to monomethylamine, dimethylamine, or the two different type of compound that will be formed. And they undergo the reaction S. S is nothing but methanogenesis from methylamines. That leads to the production of methane. So the higher order compounds are getting converted by various reactions to methane. So this is what is the decomposition that is happening in the soil organic carbon in the high Arctic peat soils. And we also talked about the Wolff cycle which is involved. So what is a Wolff cycle? It is basically another methanogenic pathway used by archaea. Here the archaean takes hydrogen and carbon dioxide and cycles them through the various intermediate to create methane. So some of the enzyme details are given here. All these are basically enzymes A, B, C, D, E, F up to J. All these enzymes basically are involved in the conversion of this overall process. So here various chemical reactions are given. We will be looking at each step. Basically CO2 is introduced in the first step. So formyl methyl methanofuran dehydrogenase is the first enzyme that is involved that leads to this particular product, and then this is nothing but formyl methyl methanofuran molecule. So this is furan and this is the formyl methyl group is present here. So this is nothing but formyl methanofuran molecule, and this undergoes various steps to give finally methane. Hydrogen is produced here and methane is produced here. So the carbon dioxide is getting converted through all this process to methane. So that is what is called the Wolff cycle. So we will be looking at each chemical reaction that are happening here. So the reduction of carbon dioxide occurs by a cycle where the C1 group is passed in a bucket brigade fashion. So what do you mean by bucket brigade fashion? Because in the construction, what people will do is like there are in the olden days, the labor was very high. So that means the people were a lot. So the bucket brigade is mainly called as some bricks will be transferred from one person to another person in a bucket, and this will go like a chain. And every time they will be carrying only one or two bricks, and they will be transporting from the place where it is stored to the place where the construction actually takes place. So that is what is called the bucket brigade. And similarly, we will also be looking at a similar type of reaction. That means one step or one transformation takes at a single stage. So that is what this entire process is called bucket brigade. And here the C1 group, that is one carbon, is bound to the coenzyme and it is subsequently reduced. That means it is transferred from one place to another place, and during the process it is getting reduced. So we start with the carbon dioxide. The first stage is basically the CO2 fixation. So the gaseous carbon dioxide is now fixed to the molecule. So methanofuran structure is given here, and this methanofuran gets the carbon dioxide. One carbon is added to this methanofuran. That is what we end up in the formyl group. So the carbon is introduced or fixed to the substrate. And then the next stage, CHO group transfer takes place from the MFR to MPT. That is tetrahydromethanoterene is the compound. The H4 MPT is the one which takes away that formyl group transfer happens here. So you have, when we look here, we have the formyl group. That formyl group is missing in this particular one. So that is how the first transfer takes place. And then the next stage is the Schiff's base formation happens. That leads to the double bonded compound, and further another, this is methynyl functionality is formed here, which undergoes further reduction to methylene group, and this further undergoes reduction to methyl group, and finally it is transferred to a cofactor, and then this methyl group is finally converted by the methyl reductase system to methane. So that is the overall reaction. So the first one is the carbon dioxide fixation, then it is followed by N-formylation reaction, then a Schiff's base formation is given. We will be looking at each step elaborately. And this Schiff's base formation is followed by the immunoreduction. So this is the Schiff's base that is being formed, and this is the N-formyl group transfer that actually happens. And then the imine reduction means this double bond, C double bond N, is reduced to Cn as shown here as a single bond, and then followed by another reduction. So here the ring opens up, so that is the reason that is the next reduction that is happening. Then finally the transfer of this methyl group actually takes place, and then this methyl group is released as a methane molecule. So this is how the carbon dioxide, this carbon is converted into methane in the methanogenesis. So here are the things like H4 MPT is the tetrahydromethanoterene. So this compound structure is shown here. The first stage is the formylation reaction, that is N-formylation reaction. So this is the fifth nitrogen. So the formylation happens on this particular nitrogen, and the next stage is we have an amine, we have a aldehyde group. So this aldehyde and the amine reacts to give the Schiff's base. So that is the next stage which happens with the loss of water molecule. Then 2 electron transfer takes place. That is nothing but the reduction of this imine bond is actually happening, which results in the amine, a C nitrogen single bond is formed, and then followed by another 2 electron reduction leads to the release or the breakage of this CN bond, gives the methyl group, and this methyl group is now then transferred to the enzyme, and from the enzyme it is getting converted to the methane as a final methyl reductase converts the methyl group into the methane. So that is how the whole cycle ends. And the next set of reactions are conversion of propionate to methane. So first step, because this also we had seen in the degradation of organic soil, how the reaction actually takes place. So the propionate fermentation reaction is the one here. The propionate salt reacts with water to give acetate salt and formate salt, and 2 hydrogen atom and H plus ions are being produced. And then the next step, another step is the ethanol fermentation reaction, because these are all the reactions we had seen in the degradation products. So what is the ethanol fermentation reaction? Ethanol reacts with bicarbonate anion to give the acetate anion plus H plus ion, and 2 propionate is formed plus 3 water molecule. And in the case of acetoclastic methanogenesis, this reaction we had seen, this formed acetate reacts with the H plus ion to give methane and carbon dioxide. And in the methanogenesis from hydrogen and carbon dioxide, the formed carbon dioxide now reacts with the hydrogen atom to give another methane molecule and water molecule. And finally, the methanogenesis from formate. The formate is actually formed in the first stage. So this formate reacts with H plus ions and water to give methane and again carbonate salt. So this is the overall conversion of propionate to methane. And we also have the last class which are methyl reducing or methyl involved methanogenesis species. So here the conversion of methanol and methylamine to methane takes place. Methanol is getting converted to methane and bicarbonate and H plus ions and water molecule. So again another methanol reacts with the acetate to give methane molecule. Methanol reacts with hydrogen to give methane molecule. So these are all the amines, different types of amines are given here. They all react with water to give methane molecule. And finally, bicarbonate also reacts with hydrogen to give acetate salt. And this acetate salt we have actually seen here. This reacts with the methanol to give methane. So all these reactions lead to final formation of methane.